today's topic is bacterial recombination, which includes transformation and transduction. So today we are going to see what is transformation and what is transduction. So first of all, we need to know what is genetic recombination. So genetic recombination is transfer of DNA from one organism to another organism, where the transferred donor DNA will be integrated into the recipient's genetic material by various mechanisms. In that way, where genetic variation occurs within an organism, like mutation, genetic recombination contributes to genetic diversity of population. So bacterial recombination occurs total in three ways. It is transformation, transduction, and conjugation. So first of all, we will see what is transformation. Transformation is genetic recombination in which a dead fragment from a dead degraded bacteria enters a competent recipient bacteria, where it is exchanged for a piece of recipient DNA. So this transformation is a process which occurs in prokaryotes actually, where bacteria has an ability to uptake some foreign DNA which is present in its surroundings, so that where that foreign DNA will be entered into the bacterial cell and will be integrated into its own DNA. So in that way it gives change. So here you can see a donor bacterium dies and is degraded, and a fragment of dead donor bacterium binds to the DNA binding proteins of a living bacterial cell, so that this dead fragment will be entering into the living competent recipient bacteria. And finally, due to the with the help of recombinant A protein, this donor DNA will be integrating into the recipient DNA. So in that way genetic diversity takes place, and where it leads to genetic recombination in case of bacteria. So this is one of the methods in case of prokaryotes. It is especially in bacteria. So this is where exchange is completed. So transformation with DNA fragments, and this is transformation with a plasmid. There are two types here showing, and one where fragments will be integrating into the living bacterial cell, and one more where plasmids, because bacteria has a property to uptake everything that is present outside their body cell. Even if there is any plasmid outside the bacterial cell, then they will uptake that plasmid into within its cell. So this is also one kind of transformation. So next method is transduction. So transduction occurs in two types. One is generalized transduction and specialized transduction. So in case of transformation, there is no such any medium to for the exchange of this genetic material, where bacterial cell can directly uptake some foreign DNA within its ambience. But in this transduction, there will be an use of a vector source. In this case, in case of transduction, there will be a bacteriophage, which is a virus that will infect this bacteria, and this virus will infect from one bacteria to another bacterium, thereby transferring its genetic material from one bacteria to another bacteria, and which leads in the promotion of genetic diversity. So here you will see here what are bacteriophages. Bacteriophages are obligate intracellular parasite that multiply inside bacterium by making use of some or all of the host biosynthetic machinery. So transduction, as I already told, it is of two types. One is generalized transduction and specialized transduction. In generalized transduction, a DNA fragment is transferred from one bacteria to another by a lytic bacteriophage, that is carrying donor bacterial DNA due to an error in maturation during lytic life cycle. In case of specialized transduction, DNA fragment is transferred from one bacteria to another by temperate bacteriophage, uh, where it undergoes through lysogenic phase. In first generalized transduction, we will observe a lytic phase of bacterium, and second one, specialized transduction, will observe lysogenic phase. We will see the further steps. So in the first generalized transduction, here there are 7 steps in this, where here you can observe the lytic bacteriophage observes to the susceptible bacteria, where here the virus is infecting the bacteria. Now, where in the second slide you see, in second step, it incorporates this viral genome into the bacterial cell, where the genome where it finally this genome will enter into the bacterial cell, and where it starts replicating. So after entering of the bacterial phage DNA into the bacteria, in the starting phase it destroys the bacterial DNA. It breaks the bacterial DNA into pieces, and at the same time, this viral bacteriophage, this bacteriophage, which is a virus, it also replicates apart from breaking the DNA of bacteria. And so here you can see uh, here the red portions in the phage particles are of own bacteriophage DNA, whereas this violet portion you can see here, that is the broken pieces of its own bacterial DNA genome. So here you can see, after the formation of these bacteriophages, some violet portions are entering into the new bacteriophages, and some are correctly placed with red portions of bacteriophage, that is its own bacteriophage DNA. So in that way, this violet portion, which is of bacterial DNA, will be entering into the viral DNA, viral cell, that is viral particles, that is first particles. Here you can see that. So now this lytic phase is arise, and where cell of bacterium will break, and you can see the release of bacteriophage particles. And among these bacteriophage particles, these are called normal bacteriophage, because this red color indicates of own DNA of bacteria of viral, whereas here the violet portion is of donor bacterium DNA, which is broken into pieces. So one piece was entered. Here you can see the piece of DNA entering into the bacteriophage, and now these bacteriophages are released after the death of bacteria. And after the release of these bacteriophages, again this bacteriophage will infect new bacterial cell, where it injects its genome into the bacterial cell. Now this donor bacterial DNA, which is injected into the new bacterial cell, will be incorporating into the recipient bacterium's nuclei. Here you can see the violet portion integrating into the recipient bacterium. So here in this way, the bacterial DNA is exchanged from the donor bacterium to the recipient bacterium, and where genetic transformation, uh, transduction occurred, and where genetic recombination occurred in this process. And the second one is uh, specialized transduction. So similar with the generalized transduction, in case of specialized transduction, there will be a little difference in this, where here instead of after a bacteriophage DNA, when it entered into the bacterial cell, it doesn't break the bacterial DNA immediately. What it will do, this bacterial phage genome will directly incorporate into the bacterial uh, DNA, which finally form as a prophase. Prophase is nothing but the viral DNA, bacteriophage DNA after entering into the bacterial cell will be incorporating into the bacterial DNA, but it won't break the bacterial DNA into pieces, and this is called prophase. And now this prophase will be multiplying into several copies here within the bacterial cell, and after at some this is called lysogenic phase. This phase is called where without breaking the bacterial DNA, this phase is called lysogenic phase. But at some point in future, if there is some favorable condition, again this fast DNA will separate from the original bacterial DNA, and after at the time of separation, you can see here fast DNA will be separated. Along with that, some donor DNA nucleide will also come along with this fast DNA, and this both DNA will be included into the fast particle, because this uh, there after the multiplication of several copies of the DNA. So now you can see the pink portion as well as violet portion, where the pink portion is of fast DNA and violet portion is of new bacterial DNA. Now this entire genome entered into the bacteriophage, and several bacteriophages are formed in the each bacterial cell. And at this phase again, lytic phase starts, where bacterial cell will be break open, and uh, it will release the fast particles, and these fast particles now, which are having both fast DNA as well as bacterial DNA, will infect the new bacterium here. Now the bacteriophage observes to the receptor and injects it. Here you can see the bacteriophage injecting its genome, where it contains both fast DNA as well as old uh, old bacterial DNA. Now these two portions will be incorporating into the new bacterial DNA, where now at the uh, uh, end of the stage, it will be having three different DNAs, that is one fast DNA, second one donor DNA, along with the DNA, recipient bacterium DNA. So this is the process of specialized transduction. These are the various stages of uh, genetic recombination in case of bacteria.